Welcome to Chapter 14, Glycolysis, Gluconeogenesis, and the Pentose Phosphate Pathway. In this lecture, we will be discussing part 3 of this chapter. Besides understanding gluconeogenesis and the pentose phosphate pathway, we will also be discussing the fates of pyruvate under anaerobic conditions. Glycolysis is linked to other metabolic pathways. Now, as you know, glycolysis is the process of converting glucose to pyruvate. And in this process, 2 ATP and 2 NADH are formed. The NADH that is formed enters into what is called as the electron transport chain in the mitochondria. The pyruvate, however, enters what is called as the Krebs cycle. And this is how pyruvate gets converted to small molecule like carbon dioxide, and as a result leads to formation of more ATP molecules. Now let us discuss in detail as what other pathways can pyruvate participate in. Again, the fates of pyruvate are summarized in this specific slide. 2 molecules of pyruvate are formed from glycolysis of a single molecule of glucose. Lots of energy is still left in these molecules, right? The pyruvate is not the last stop of glycolysis. Under aerobic conditions, in the presence of oxygen, pyruvate starts the TCA cycle, or citric acid cycle as it is called. Under anaerobic conditions, pyruvate undergoes fermentation. There are 2 types of fermentation. The first one is conversion of pyruvate to ethanol and carbon dioxide, and the second type is the conversion of pyruvate to lactate under anaerobic conditions. During anaerobic glycolysis, generation of energy or ATP happens without consuming oxygen or NAD+. There is no net change in oxidation state of the sugars. Reduction of pyruvate to another product happens. This regenerates NAD+ for further glycolysis under anaerobic conditions. The process is used in the production of food from beer to yogurt to soy sauce. It's a huge industry. Ethanol is the reduced product in ethanol fermentation. In this process, pyruvate is first converted to acetaldehyde by the enzyme pyruvate decarboxylase, and the acetaldehyde that results from this reaction is then converted to ethanol by alcohol dehydrogenase. It's a 2-step reduction of pyruvate to ethanol. However, humans do not have this enzyme pyruvate decarboxylase, and ethanol fermentation mostly happens in yeast and other microorganisms that ferment glucose to ethanol and carbon dioxide. We also express alcohol dehydrogenase, not to convert acetaldehyde to ethanol, but for ethanol metabolism. If you remember, the way ethanol is metabolized in our body is by converting it to acetaldehyde, and then acetaldehyde gets converted to acetic acid by acetaldehyde dehydrogenase. In yeast and microorganisms, carbon dioxide produced in the first step is responsible for carbonation in beer or dough rising when baking bread. Both steps require cofactors. For example, pyruvate decarboxylase requires a cofactor that is called as thiamine pyrophosphate and magnesium. Alcohol dehydrogenase, on the other hand, requires NADH as a cofactor and zinc as a metallo cofactor. Thiamine pyrophosphate, or TPP, is a cofactor that is derived from vitamin B1. It is an important cofactor that is used by enzymes to cleave bonds next to carbonyls. The thiazolium ring in TPP, as shown here, stabilizes carbanion intermediates by providing electrophilic structure into which the carbanion electrons can be delocalized by resonance. Let's look at an example. The business end of TPP is shown here. It's the thiazolium ring, and the thiazolium ring can ionize into this TPP carbanion, and the carbanion can attack the carbonyl carbon of pyruvate. Once it attacks, it forms an adduct like this, and the adduct, as you can see, has a structure wherein the nitrogen, which is positively charged, can act as an electron sink. And because of that reason, the carboxyl group can push the electrons in, and the bond electrons can then push in between the CC bond, and this double bond can then push into this nitrogen, which is an electron sink. The result is a decarboxylation reaction, and it is facilitated by electron delocalization into the thiazolium ring, as seen here. And the thiazolium ring is now resonance stabilized, and it is stabilizing a carbanion. Once it is resonance stabilized, it can then abstract a proton, and once the proton is abstracted, this specific OH bond can then move in to get released from TPP, and the product acetaldehyde then is eliminated from the enzyme active site. So this is a typical mechanism for enzymes that use TPP as a cofactor. As you can see, TPP makes an adduct with specific compounds that forms a carbanion and stabilizes it. In lactic acid fermentation, pyruvate is converted to L-lactate, and this conversion is catalyzed by the enzyme lactate dehydrogenase, and it uses the cofactor NADH. When animal tissues cannot be supplied with sufficient oxygen to support aerobic oxidation of the pyruvate and NADH produced in glycolysis, NAD+ is regenerated from NADH by the reduction of pyruvate to L-lactate. So why do you need to regenerate NAD+? Because if you regenerate NAD+, it can re-enter the glycolytic pathway and get converted back to NADH. So, and the result is that it can then produce more ATP. Some tissues and cell types, such as erythrocytes, which have no mitochondria and as a result cannot oxidize pyruvate to carbon dioxide, produce lactate from glucose. This means that in erythrocyte, glucose gets converted to lactate via lactate dehydrogenase pathway. And this is a reversible process. During strenuous exercise, lactate builds up in the muscle, generally less than 1 minute. The acidification of muscle prevents its continuous strenuous work. The lactate can be transported to the liver and converted to glucose there. Requires a recovery time, high amount of oxygen consumption to fuel gluconeogenesis and restores muscle glycogen stores. And that is the function of a lactate. And hence, lactic acid fermentation is an important process that happens in humans as well. Let us ask this question one more time. Why lactic acid fermentation? In the absence of oxygen, pyruvate cannot participate in aerobic process of ATP production. What do I mean by aerobic process? Again, pyruvate enters TCA cycle and gets converted to carbon dioxide. That involves aerobic oxidation of pyruvate to carbon dioxide. When that option is not available, ATP production can only proceed via glycolysis, and in this case, anaerobic glycolysis. Now, for this process to happen, one needs to replenish NAD plus stores, because the NADH, two NADH molecules that gets generated from glucose can only be converted to NAD plus via this lactate fermentation process. The second process that need to happen here is to export pyruvate to promote continued glycolysis. Glycolysis occurs at elevated rates in tumor cells. Most tumor cells grow under hypoxic conditions, that is, with limited oxygen supply. This is because, at least initially, tumor cells lack the capillary network to supply sufficient oxygen. Tumor cells that are located more than 100 to 200 micrometer from the capillaries that are nearest to them must depend on glycolysis alone for energy. So tumor cells increase anaerobic glycolysis pathway by expressing more lactate dehydrogenase. Now, what happens is that when tumor cells needs glycolysis process, it has to overexpress many different enzymes in the glycolytic pathway. This is done by the hypoxia-inducible transcription factor, or HIF1. This is a protein that acts at the level of mRNA synthesis to stimulate the production of at least 8 glycolytic enzymes, including glucose transporters that take up more glucose. And hence, compounds that inhibit key steps in glycolysis can kill cancer cells by limiting energy production. And hence, cancer cells die before even getting to angiogenesis, or before even getting oxygen from capillaries that grow towards them. What are the sources of glucose? Glycogen and starch need to be broken down before they can enter glycolysis. Glycogen phosphorylase uses a phosphate to attack the O-glycosidic bond of glycogen to produce glucose 1-phosphate. Most carbohydrates we ingest are polymers, and not all are polymers of glucose. These polymers are degraded by digestive enzymes into monosaccharides. Non-glucose monosaccharides are enzymatically converted to glucose or other components of the glycolytic pathway. The image shown here describes in detail the sources of glucose. Glucose can be obtained from sucrose. It's just sugar. And sucrose, the enzyme sucrase converts sucrose to glucose and fructose. Glucose can then enter glycolysis via hexokinase pathway. Glucose can also be obtained from dietary glycogen and starch, which gets converted to glucose 1-phosphate, which then gets converted to glucose 6-phosphate by phosphoglucomutase, and glucose 6-phosphate can then enter the glycolytic pathway. Fructose can be converted to fructose 6-phosphate by hexokinase as well. Disaccharides usually are hydrolyzed. Lactose can get converted to glucose and galactose. Fructose, galactose, and mannose enter glycolysis at different points. Mannose can be converted to mannose 6-phosphate, which can then get converted to fructose 6-phosphate by mannose isomerase. Galactose can be converted to glucose 1-phosphate via these metabolism. But the real question is, what happens if you lack functional enzymes? The first digestive defect is lactose intolerance. This is common among adults of most human populations except those originating in Northern Europe and some parts of Africa. And this digestive effect results from the lack of digestive enzyme lactase to break down the disaccharide lactose. Without this intestinal lactase, lactose, as shown here, cannot be completely digested and absorbed in the small intestine. And it passes into the large intestine where bacteria convert it to toxic products and cause abdominal cramps and diarrhea. The problem doesn't stop there. It is further complicated because undigested lactose and its metabolites increase the osmolarity of the intestinal contents, favoring retention of water in the intestine. Conversion of galactose to glucose 1-phosphate proceeds through these reactions, and they're catalyzed by various enzymes starting with galactokinase. Galactosemia is a defect in any of the three enzymes that convert galactose to glucose. This can lead to toxic levels of galactose 1-phosphate. This results in liver and kidney damage that can progress to seizures, brain damage, and infant mortality, up to 75% of them. This brings us to our next topic, gluconeogenesis. What does gluconeogenesis mean? It means formation of new sugar. This process is necessary during times of low nutrition, during fasting, and after vigorous exertion because stored glycogen is depleted. And during an exercise, this glycogen is converted to glucose, which is used for glycolysis. Gluconeogenesis is energetically costly, but it is necessary. This process occurs primarily in the liver, and it is almost the reverse of glycolysis. Why do we need gluconeogenesis, you may ask? And it is a really good question. Glucose is near universal as fuel and a building block. Many organs rely solely on blood glucose for energy. Brain, on the other hand, requires about 120 grams of glucose each day. That is a lot of glucose. Glucose stores can be rapidly depleted during high activity or prolonged fasting. Glycogen stores in liver and muscles need to be replenished after activity. And finally, mammals cannot convert fatty acids to sugars. And these are the reasons why we need the process called gluconeogenesis. And it is literally nothing but formation of glucose from all the byproducts that were originally formed via glycolysis. Here is the major difference between glycolysis and gluconeogenesis. Glycolysis, as I said, is converting glucose to pyruvate. And in this process, ATP is produced. Whereas gluconeogenesis is the process of converting pyruvate into glucose. And in this process, ATP has to be invested. And hence, it's energetically costly. Glycolysis occurs mainly in the muscle and brain. Gluconeogenesis, on the other hand, occurs mainly in the liver. We have seen this table before. And the reason why I'm showing this table again is to emphasize 2 points. The first one is that there are 7 steps in black that are kept at near equilibrium. As you can see, 1, 2, 3, 4, 5, 6, 7. On the other hand, the 3 red steps that are in red that are effectively irreversible reactions in glycolysis. And because of this, they need to be bypassed if gluconeogenesis were to happen, because gluconeogenesis is going from pyruvate back to glucose. There are 3 major differences in the reactions between glycolysis and gluconeogenesis. And that is to do with the reason that I explained it to you in the previous slide, because there are 3 irreversible reactions in glycolysis. So those 3 reactions need to be replaced. And that's what happens in gluconeogenesis. The first difference is that phosphoenol pyruvate is made from pyruvate via 2 enzymatic reactions. Pyruvate carboxylase converts pyruvate to oxaloacetate, and this happens in the mitochondrion. As you can see, pyruvate gets converted to oxaloacetate. Now, if cytoplasmic NADH levels are low, what happens is that oxaloacetate gets converted to malate in mitochondrion, where NADH in mitochondrion is utilized. This malate then goes outside the mitochondrion to the cytoplasmic side, where it gets converted to oxaloacetate by cytosolic malate dehydrogenase. And as a result, NADH is produced. If the cytoplasmic NADH levels are high, then the oxaloacetate that is produced within the mitochondrion moves to the cytoplasmic side, and oxaloacetate gets converted to phosphoenol pyruvate by the action of phosphoenol pyruvate carboxykinase. Cytoplasmic malate is oxidized to oxaloacetate and converted to phosphoenol pyruvate as well. And this happens, again, as I said, when the cytoplasmic NADH levels are low. The second difference is the conversion of fructose-1,6 bisphosphate to fructose-6 phosphate happens in the presence of an enzyme, fructose-1,6 bisphosphatase. And it is a magnesium-dependent enzyme, and it hydrolyzes the C1 phosphate in fructose-1,6 bisphosphate. And it produces phosphate, not ATP, from ADP. The third difference is the conversion of glucose-6 phosphate to glucose is catalyzed by glucose-6 phosphatase. Again, hydrolysis of C6 phosphate produces phosphate and not ATP from ADP. So these 2 reactions are expensive in the sense that they are not producing any ATP. The third enzyme, glucose-6 phosphatase, carries out this reaction in the endoplasmic reticulum. If you consider the differences between glycolysis and gluconeogenesis, they are opposing pathways that are both thermodynamically favorable. They operate in opposite directions. The end product of one is the starting compound of the other. Reversible reactions are used by both pathways. As I said before, there are 7 reactions that are reversible and are used by both glycolysis and gluconeogenesis. And those reactions are kept at near equilibrium, and that is for a reason. Irreversible reactions of glycolysis must be bypassed in gluconeogenesis because they are highly thermodynamically favorable and regulated. There are different enzymes in different pathways. differentially regulated to prevent a futile cycle. And so, when we talk about the cost, how expensive is gluconeogenesis? 2 pyruvate molecules in presence of 4 ATP and 2 GTP and 2 NADH gives rise to glucose with 4 ADP, 2 GDP and 6 phosphate along with 2 NAD+. It is really expensive because you're utilizing 4 plus 2, 6 ATP equivalents. 1 GTP is equivalent to 1 ATP. So 2 GTP is equivalent to 2 ATPs. So 4 plus 2, 6. And remember, 1 NADH is equivalent to 2.5 ATPs. So 2 NADH is equivalent to 5 ATPs. So 5 plus 6 is 11 ATP molecules are required to convert 2 pyruvate molecules to 1 glucose molecule. Again, that's the cost. And physiologically necessary because brain, nervous system, and red blood cells generate ATP only from glucose. And hence, glucose levels need to be refurbished. Allows generation of glucose when glycogen stores are depleted. And this is during starvation, during vigorous exercise, and it can generate glucose from amino acids, but not fatty acids. This is an important point. What about precursors of gluconeogenesis? Animals can produce glucose from sugars or proteins. Sugars involve pyruvate, lactate, or oxaloacetate. Proteins from amino acids that can be converted to citric acid cycle intermediates or glucogenic amino acids. However, animals cannot produce glucose from fatty acids. The product of fatty acid degradation is acetyl-CoA, and there cannot have a net conversion of acetyl-CoA to oxaloacetate. And animals cannot do this conversion, while plants, yeast, and many bacteria can convert acetyl-CoA to oxaloacetate, thus producing glucose from fatty acids. So here are the two alternate fates for pyruvate. If you consider this diagram, pyruvate can be converted to oxaloacetate, which enters gluconeogenesis to produce glucose. Alternatively, pyruvate can enter the TCA cycle or the citric acid cycle by converting to acetyl-CoA and thus to energy. Now, it can be stored as glycogen or generate NADPH via pentose phosphate pathway. This is an alternate pathway to glycolysis. Pyruvate can be source of acetyl-CoA, like I said, store energy as body fat, or make ATP via citric acid cycle. Acetyl-CoA stimulates glucose synthesis by activating pyruvate carboxylase. Let us begin our discussion on pentose phosphate pathway. Glucose-6 phosphate is also used to synthesize pentose sugars. And where do pentose sugars get utilized? Pentose sugars, as you may remember, are used in making nucleotides, right? DNA and RNA. Now, conversion of glucose-6 phosphate to pentose sugars involve a 4-step reaction that is coupled to the oxidation of NADPH. The main products are NADPH and Ribulose-5-phosphate. So Pentose Phosphate Pathway gives two products, NADPH and Ribulose-5-phosphate. NADPH is an electron donor. It's used in reductive biosynthesis of fatty acids and steroids and repair of oxidative damage. Ribulose-5-phosphate, on the other hand, is a biosynthetic precursor of nucleotides used in DNA and RNA synthesis or synthesis of some coenzymes. The two phases of pentose phosphate pathway is simplified in this specific image. In the oxidative phase, glucose-6 phosphate gets converted to 6-phosphogluconate while producing NADPH, and 6-phosphogluconate gets converted to ribulose-5-phosphate, producing another molecule of NADPH. Ribulose-5-phosphate that then gets converted to ribose-5-phosphate, which is then utilized in making nucleotides, coenzymes, DNA, and RNA. In the non-oxidative phase, recycling of 6 molecules of ribose or ribulose-5-phosphate in this case into 5 molecules of glucose-6 phosphate takes place. And the reason for this non-oxidative phase, which is catalyzed by trans-ketolase and trans-aldolase, is to continue the production of NADPH and converting glucose-6 phosphate into carbon dioxide in glycolysis. So 2 different phases, non-oxidative and oxidative phase, complete the pentose phosphate pathway. Let us first consider the reactions in oxidative phase of pentose phosphate pathway. In the first step, glucose-6 phosphate is converted to 6-phosphogluconodelta-lactone. This is catalyzed by glucose-6 phosphate dehydrogenase. And in this process, a molecule of NADPH is generated. The product, as you can see, has this lactone ring and hence gluconodelta-lactone. In the second reaction, another enzyme, lactonase, converts 6-phosphogluconodelta-lactone to 6-phosphogluconate. And this is literally a hydrolysis reaction. And as you can see, glucose now has a carboxylate and hence 6-phosphogluconate. 6-phosphogluconate then gets converted to D-ribulose-5-phosphate by the enzyme 6-phosphogluconate dehydrogenase. And in this process, 1 molecule of NADPH is generated. Ribulose-5-phosphate is then converted to D-ribose-5-phosphate by the enzyme phosphopentose isomerase. Ribulose-5-phosphate has 2 fates. It can either be converted to D-ribose-5-phosphate, which is then used for making nucleotides, or ribulose-5-phosphate can be used to generate xylulose-5-phosphate. Xylulose-5-phosphate is an important intermediate for the non-oxidative pentose phosphate pathway. The non-oxidative phase regenerates glucose-6 phosphate from ribose-5-phosphate, and it is used in tissues requiring more NADPH than ribose-5-phosphate, such as liver and adipose tissues. No ATP is used in the non-oxidative phase. Complete conversion of ribose-5-phosphate to glucose-6 phosphate requires 6 molecules of ribose-5-phosphate to make 5 glucose-6 phosphate molecules. And as you can see, the reactions that are involved in this process is carried out by 2 different enzymes, trans-ketolase and trans-aldolase. And actually, there are 2 different trans-ketolase reactions involved in this. And in this case, xylulose-5-phosphate gets converted to glyceraldehyde 3-phosphate and acetoheptulose-7-phosphate, which then gets converted to fructose-6 phosphate and erythrose 4-phosphate by trans-aldolase. Fructose-6 phosphate can be converted to glucose-6 phosphate, whereas erythrose 4-phosphate can again get converted to fructose-6 phosphate by the action of xylulose-5-phosphate in the presence of trans-aldolase reaction. So this set of reaction is pretty standard, and happens in this reaction is that the 5-membered xylulose 5-phosphate and another 5-membered ribose 5-phosphate gets converted to a 7-membered and a 3-membered sugar by transketolase. Whereas the 7-membered sugar in combination with the 3-membered glyceraldehyde 3-phosphate gets converted to erythrose 4-phosphate, which is a 4-membered sugar, and a 6-membered fructose-6 phosphate by transaldolase. Now, it is worth noting that transketolase uses a thiamine pyrophosphate cofactor, whereas a transaldolase uses a lysine to make a Schiff's base so that it can initiate cleavage of a 7-membered sugar to a 4-membered and a 6-membered sugar. NADPH can regulate partitioning into glycolysis versus pentose phosphate pathway. When NADPH is forming faster than it is being used for either biosynthesis of fatty acids or in glutathione reduction in removing oxidative stress, the concentration of NADPH rises and inhibits the enzymes that catalyzes the oxidative pentose phosphate pathway, and especially the first enzyme in the pentose phosphate pathway. As a result, more glucose-6 phosphate is available for glycolysis to form ATP. Glucose-6 phosphate dehydrogenase deficiency can be fatal in case of high oxidative stress. This enzyme catalyzes the first step in the pentose phosphate pathway. It converts glucose-6 phosphate to 6-phosphogluconodelta-lactone. And in the process, it converts NADP to NADPH. The NADPH resulting in this process is utilized by glutathione reductase, and it uses it to reduce glutathione. And this is used by glutathione peroxidase to convert hydrogen peroxide to water and to prevent other oxidative damages to lipids, proteins, and DNA. In the absence of NADPH that comes out of this reaction, oxidative stress increases, and as a result, this can lead to death.